I'm Daniel Levine, and this is the Bio Report.
The genomics revolution promised to unravel diseases and lead to treatments that addressed their root causes. In reality, says Mo Jain, your zip code remains a better predictor of how healthy you'll be over the course of your life than your genetic code does. That's because, except for monogenic diseases, etiology tends to be far more complex than the identification of a single gene. Sapient Bio is using its AI-driven multi-omics platform to advance the discovery and development of precision medicines. We spoke to Jain, Chief Scientific Officer of Sapient, about how its technology bridges the gap from discovery to clinical development, accelerates drug development timelines, and expands opportunities to drug previously undruggable targets. Mo, thanks for joining us.
Thanks so much, Danny. Wonderful to be here today.
We're going to talk about drug discovery, Sapient, and how its AI-driven multi-omics technologies advance the discovery and development of precision medicines. You're a physician scientist. You had an accomplished academic career. How did your work at UCSD lead to the founding of Sapient?
Yeah, absolutely. I'm going to, if you don't mind, Danny, I'll take a step back and go pre-UCSD, if you will, just so you understand the trajectory and how we got to this point. And so, and as you mentioned, I was an academic. I was trained as an MD, PhD. I was a clinical cardiologist and absolutely adored the pleasure of taking care of patients and that privilege. And one of the frustrating sort of aspects of medicine is the longer you do it, the more you realize how little you really know. And I suspect that's the case for most technical fields, but in medicine, it certainly is challenging when you have patients who ask me, why did I get sick? Or how do you know this drug is going to work for me? Or how do you know this won't work for me? Et cetera, et cetera. And as you go through that process, through medical school and beyond, you learn the typical answers that are in the textbooks. But the challenge or the benefit of practicing in Boston is oftentimes you get these academics who are admitted to the hospital, and they've heard the textbook answer and they're looking for something further. And when you really sort of have those conversations with your patients, it becomes really abundantly clear, not only to yourself, but to the patient, just how little we understand. Now, if we think back to the human genome, the great promise of the human genome from the early 2000s was this was going to provide the information. This was going to close the gap. This was going to tell us who exactly is going to develop what disease and how best to treat them. And in certain areas of medicine, that promise has been a reality. What I would say as a whole, though, is one of the real sort of learnings over the last 2 decades with, 1 million genomes being released publicly, et cetera, is that we can only explain a minority fraction of disease risk, what we call population attributable disease risk. It's typically on the order of 15 to 20% for most common diseases, whether it be heart disease, cancer, neurodegeneration, lung disease, GI and liver illness, autoimmune disease, et cetera. So there's still 80, 90% that's unknown. What that means is it's like trying to solve a puzzle when you only have 10, 20% of the pieces and you don't know what the final puzzle image even looks like. It's virtually impossible. Mathematically, we call this underdetermined. We just don't have enough information or enough equations to constrain the underlying answer. And so the solution space becomes essentially infinite, and it becomes an impossible problem to solve. And so I got very interested early on in how do we fill that other 80, 90% gap in understanding human disease. In hindsight, you could say that this was somewhat obvious, Danny, in that if you look statistically, in the simplest of terms, your zip code has a better predictor for how healthy you're going to be over the course of your life than your genetic code does. And that's just a mathematical fact again. And so This suggests that there's many factors that extend beyond your genome. And remember, your genome is set for the most part for the moment of conception and doesn't change over the course of your life. So there's many other factors, how you live your life, everything you eat, drink, smell, smoke, how one organ system or one cell type communicates with another cell type. All of that dynamism, those dynamic processes that are not captured in genomics. And so that's what I became very interested in. How do we measure that? How do we capture that information? And then how do we leverage it for the promise of precision guided therapeutics, both from a development perspective and a deployment perspective? And that's where I spent the last 2 decades of my career. The long and the short of it is we began working extensively in a number of technologies, particularly those which are called mass spectrometry. These are complex devices that can take complex specimens such as blood or tissue or tumors or cells or animal model samples and separate the thousands of components that are in those complex specimens and quantitate them. And in doing so, in the same way we do haplotype analysis with genomics, we can begin now at scale to understand these other factors and fill in those 80, 90% of a missing gap.
Oh, was there a moment when you realized that what you were doing could be applied at a large scale for industry? When did it go from just being an academic curiosity to something that was a business?
Absolutely. So this happened about 8 years ago, Daddy. And the progression, as is often the case in science and medicine, is we built these devices that for the first time could measure thousands of things in complex specimens. And we could do this at scale, meaning from 1,000 to 10,000 samples at a time, not knowing at all what we were going to find when we generated the data. And so I can remember back vividly to approximately 8 years ago when we generated the first data set and then we sat down and started analyzing it, again, not knowing what we would find. And it was absolutely remarkable, the things we were able to predict about people. So for the first time, we could predict who was going to develop what disease over a period of time, what drug they were going to respond to, who was going to have an adverse event, the prognosis, meaning for any given disease, who was going to do well and who was going to do less well. And it was absolutely clear that extending these types of measurements to large numbers of individuals had enormous promise. And through that process, we came into contact with a number of large pharma companies, large international organizations, including the Bill and Melinda Gates Foundation. And many of these individuals came to us and said, hey, we have this drug, we have this disease, we're very interested in understanding it. Genomics is not providing the full information. Can we use your mass spec systems here to help us understand what's going on? How the drug may be working, who's going to respond, how do we treat them at the earliest point, et cetera, et cetera. And so that's really what gave rise then to Sapient. It was a number of conversations with large pharma companies who said, this is something you need to make available to the world so we can develop drugs faster. So approximately 6 years ago, we took the plunge and we launched Sapient, again, not knowing at all how this was going to turn out and what this was going to look like in the market. And I'm happy to say, Danny, that since then it's been largely upwards and to the right. And we've been able to have more and more adoption of these technologies as it's become really obvious and apparent to the world that we need to develop drugs faster, cheaper, that are more effective.
Sapiens says it's redefining how biology is measured and interpreted. What does that mean in practical terms?
Yeah, yeah, it's really interesting when you think about our understanding of human disease. And it's changed substantially over the last 40, 50 years. But again, we still understand such a small component of the underlying complex system. On the order of a couple percentage. And I hate to sort of reuse the puzzle analogy, but it's absolutely clear here that if you're trying to solve a puzzle and you only have a couple of the pieces, it just doesn't work. And this is obviously why drug development is fraught with failure. Even under the best conditions, 90% of therapeutics that enter into clinical trials end up ultimately failing and not receiving FDA approval over that period, whether they fail early on in phase 1 or 2 or later on in phase 3. And so this is one of the few professions that, despite massive improvements in underlying technology, there continues to be an extremely high failure rate. And it's clear that that's not sort of apparent or not is not acceptable, if you will, that we need to do better for the sake of the universe. And what this means is now by extending our ability to measure things, we can understand disease processes much better. We can understand what the targets should be. How do we actually modify disease risk? What are the earliest time points at which we can intervene before the system is truly broken? And so we could prevent the system from breaking. How do we know what is the actual best therapeutic for a particular individual and their particular disease process? How do we best deploy therapeutics to make them maximally effective? And so all of those key questions are answered by data. And we now have, for the first time ever, the ability to generate that key data at scale in large systems so we can answer these questions.
There's been a lot of talk about precision medicine, but in some ways it's been rather one dimensional with a focus on either a single gene or a biomarker, a protein. What's the potential to realize a vision of precision medicine with a multi-omics approach?
Yeah, absolutely. It's a great question, Danny. And when you can think about this, when a single gene measurement works, we have enormous success. It's the reason that rare disease therapeutics have exploded over the last decade. Not only from a regulatory perspective are there incentives, but really from a drug development perspective, we have much higher success rates when we know exactly what's broken in the system and we can actually measure it. Now, the challenges for most common diseases, and these are the diseases that the vast majority of people in the Western world are succumb to, whether it be cancer, heart disease, neurodegeneration, liver, lung, et cetera, et cetera. These are not caused by a single gene or a single process. They're multidimensional. Measuring only one gene, measuring only one RNA, measuring only one protein doesn't provide the information. You need to be able to measure these things at scale and once again understand what system has gone awry. Now, in the simplest terms, Danny, you could think of it as that the way we've defined disease is really based upon a terminal pathology, meaning we know that you start at a normal stage and we know what the system looks like when it's broken. And when the system is broken, we then say you have heart disease or cancer or neurodegeneration or dementia, or whatever the diagnosis may be. The challenge is we don't know by that end pathology how you went from point A to point B. We don't know the pathways that were dysregulated that allowed that individual to develop the disease, to go from the normal to the abnormal state. The challenge with therapeutics is therapeutics don't target the end pathology, they target the path or the mechanism that one went from normal to abnormal. And so if you diagnose disease based upon end pathology, but your drugs treat a pathway that went before that from a normal to an abnormal state, you could see where the mismatch occurs. And the simplest example I can give is, 2 decades ago or 3 decades ago when I was in medical school, we used to define lung cancer based upon its pathology, what it looked like under a histologic slide. Is it small cell lung cancer, large cell, squamous cell, et cetera, et cetera? In 2004, with the real discoveries of EGFR and KRAS, it now went to KRAS positive or EGFR positive lung cancer. And if we look today, there's over 2-3 dozen subtypes of lung cancer. And these are based upon the actual genomic pathology that gave rise, that allowed the disease process to occur, that went to once again from normal to abnormal. And each of these is treated by a different therapeutic. And so that granularity in understanding disease is really the key to precision medicine. It's not how you're different than I am different, Danny. It's not about our host. It's about how your disease process is different than my disease process. And that's really the major unlock in precision-based therapeutics.
So put that in the context of drug discovery and drug development. What are some of the bottlenecks this type of information can help seek to address?
Yeah, absolutely. So even starting from the beginning in drug development, the first question is, what's my target? Right? How do I identify the optimal target that's disease modifying, that's altered in this disease process, and whereby if I drug that target, I will alter the actual course of the disease, either reverse it or prevent it from occurring. That sounds like a very simple question. It's a really complicated one, especially when, once again, you're dealing with a complex specimen in which there's thousands of molecules or tens of thousands of molecules, many biological processes that may be altered. How do you identify what's the best target? Genomics has been extremely helpful in certain cases, particularly in the oncology space. But even there in oncology, it's only really about 15-20% of tumors have a single driver mutation that can be identified. The majority of tumors, once again, don't have a clear driver mutation where we could say this is the process that goes from normal to abnormal and cause the cancer and therefore should be drugged. What we can do now in the multi-omics space by not limiting ourselves to just the genome, but extending it to the transcriptome and then to the proteome and then to the metabolome, we can fill in those missing pieces for that 80% of missing information, or at least a majority of it. What that allows us to do now, Danny, is to say, well, in this complex tumor, it appears that this is the biological process. By measuring the full proteome, we can now say this is the biological process that's altered in this specific tumor, and therefore this should be the target, and this is where we should be drugging. Now, it not only aids in identifying that target, there's other technical aspects of drug development where multi-omics can be absolutely instrumental. And this is one of the great advances over the last several years. This is no longer limited to the early stages of drug development, but it goes from target identification and validation to chemical screening, particularly for emerging therapeutics like ADCs, T-cell engagers, PROTACs, molecular glues, et cetera. It extends now to preclinical models where essentially these multi-omics tools can answer the question is, when I put my drug into a preclinical system, into an animal model, is it doing what I think it should be doing? Is it engaging the target? Is it altering the disease biology as it should? We can use these in early translation as we think about phase 1, phase 2 clinical trials and how the drug may work in humans. And what are the best ways to monitor, once again, if the drug is doing what it should be doing and if it's altering the disease process at the earliest stages. And then ultimately in later stage clinical trials for target engagement, PK/PD, understanding companion diagnostics, selecting the ideal patients to receive therapeutics, et cetera, et cetera. So to your point, really the great advance in multi-omics is that this is no longer limited to the early stages of drug development, but now these technologies, given their advances in the last several years, can be applied across the spectrum of drug development.
So we're seeing a number of companies emerge that are focused on applying AI to accelerating drug development, others taking a multi-omics approach. What differentiates what Sapient's doing from some of its competitors?
Sure. And I could take this in two parts, Danny. The first you had mentioned for AI-based drug development, and the second you mentioned for multi-omics. And so the answers are slightly different for each one of these. When you think about AI-based drug development, the vast majority of implementation of AI as part of the drug development process has been around chemical optimization. And what that means essentially is, hey, I have my target. I believe my target is this protein or this gene. Now I need to develop a drug that engages that target. And rather than brute forcing this where I'm screening a million compounds and then figuring out what the best drug and optimizing, I'm going to use AI based upon protein structure and other types of information to identify what the best engager is of that target. Absolutely important. It's evolving very quickly. It's been slow, but it certainly is accelerating that step. That problem is that is only one step in a multi-step process. It's not helping to identify what the best target is. It's not helping to understand what's going to happen in vivo in a preclinical system or in a human. It's not enabling you to understand what are the best patients who are going to respond positively to this drug. So essentially, AI is being applied to one step in that underlying process, and therefore it's not surprising that the overall process is not being accelerated. Now, I think that's rapidly going to change as these multi-omics and data-centric tools are being applied across the entire spectrum of drug development. But up until now, this is where you've seen the majority of AI implementation. With regards to multi-omics, there's really two key sort of bottlenecks. One is the ability to generate the highest quality, most robust data that's the most comprehensive in breadth and depth possible. And that includes not only genomics, which has largely been solved by a number of organizations and a number of technologies, but beyond genomics, now extending it to proteomics and the metabolomics and lipidomics. This is where Sapient truly has deep differentiated expertise in this technology of mass spectrometry. And essentially what we can do now is take a biological specimen, whether it be blood, urine, CSF, tears, eye fluids, saliva, tumors, tissues, in a preclinical system in humans and cells in a dish, et cetera. And we can comprehensively measure for the first time the proteome, not just 1,000 proteins or 2,000 proteins, but we're hitting in complex tissues, 14, 15, 16,000 proteins, understanding protein isoforms, understanding the secondary metabolites and lipids that emerge from them. And this is, again, filling in the missing puzzle pieces. Generating the data is only the first step. Again, Danny, you have to be able to generate the best data, and then you have to be able to analyze that data at scale in a way that answers key biological questions. Just generating data for the sake of generating data is a exercise in futility, and that you'll just fill up a lot of hard drives. How we analyze that data, how we think about that data, there's a lot of nuance there. And this is where AI and other statistical tools have been absolutely instrumental in taking very complex multidimensional data and using it to answer a key biological question. And that question may be, what's the best target? What are going to be the on-target off-tissue effects of my drug? What is the best therapeutic? What is the best patient for this specific therapeutic, et cetera, et cetera. And so this is where AI is rapidly evolving and moving from just the chemical space to filling in these other key questions across the entire drug development pipeline. And in doing so, you can accelerate the entire process as a whole.
Are there particular areas of medicine that this approach is better suited for? Are there areas that you're focusing on to apply this?
Yeah, it's a great question. And I can honestly say that we don't know the answer quite yet. It's not the case that unlike genomics, that this is going to be applied only within one major therapeutic area. And if you think about genomics, the vast majority of application right now is in oncology for those 15 to 20% of tumors in which doing genomic sequence can provide quite a bit of information. And then outside of oncology, it's very, very sparse. Multi-omics, particularly proteomics, has applications across virtually every therapeutic area, whether it be oncology, whether it be neurodegeneration, whether it be autoimmune inflammatory diseases, GI illness, lung illness, et cetera, et cetera. So I think the applications will be relatively agnostic to underlying therapeutic area, but that doesn't mean it's going to work in all cases. This is not the one tool or the one ring to unite them all. But rather, it's going to be the case that there's certain situations and certain therapeutics for which is going to be extremely useful, and there's other therapeutics perhaps that this is not going to work. What I can say is our early learnings over the last several years is that those therapeutics that are highly dependent upon binding of a particular protein, particularly a cell surface protein, such as ADCs, T-cell engagers, radioligand pharmaceuticals, uh, CART therapies, both in the oncology space as well as the immunology space, are going to be transformed by these types of technologies. Our ability to identify targets is just accelerating so much quicker, and our ability to help accelerate therapeutic development is really quite profound there. I think there's other diseases such as neurodegeneration where early diagnosis and subsetting of patients is going to be the entirety of the game. And that's the key unlock for therapeutic development. And in those cases, once again, these technologies are going to be very useful, perhaps less so for drug development, but much more so for patient stratification and subsetting of disease processes.
And have you been able to discover novel signals, biological signals? And, what is the data that you're using for this? And what does it take to validate those?
Yeah, absolutely. Really good question there, Danny. And there's a couple parts to it. So the first is, what does the data actually look like? And the simplest example I can provide for you is if we take a tumor and we take normal tissue and we measure the proteins that are present in both of those and the abundance of those proteins, and then lay out this data, and there'll be 12, 13,000 proteins that typically can be measured in these biological specimens, and simply ask what's different in the tumor. You can visualize that in a variety of ways. A heat map, typically we're using what's called a volcano plot, where you're looking at the effect size, meaning how much the protein has changed and what is its statistical significance in that change between those two groups. And if you look at that for typical tumor, you'll see 2 to 3,000 proteins that are altered with tumor genesis relative to their normal tissue counterpart. Now, you can lay on top of that, Danny, what are the known and emerging drug targets as much as publicly available. And there's only a couple hundred of these that are being developed in oncology right now. And when you look at those appear somewhat at the edges, meaning that these are proteins that are differential in the tumor and that they're highly overexpressed in the tumor versus the normal tissue. What's really surprising to us now is we look at these larger data sets as we're generating them in larger, meaning not more samples, but when we're measuring more proteins, we're seeing that there's even better drug targets that are present. They're targets that are even more differential, that are highly unique for the tumor relative to the normal counterpart, even relative to those known and emerging drug targets. Now, this isn't magic. I mean, the business of science, the business of drug development is based upon observation and measurement. That's just the fundamental principle of science. And we're able to make these discoveries simply because we're able to observe more proteins. We're able to measure more proteins, more species than we could before. And when I talk about measuring more proteins, Danny, what I mean is not only asking, is this protein altered in the tumor versus the normal, but what's the actual isoform, what we call the proteoform that's altered. And what we're finding is that the protein as a whole may be 20, 30, 40% different, but there's a particular isoform of that protein, either a splice variant of that protein, a translational mismatch variant, what we call a post-translational modification, meaning there's a methyl group or a glycosylation group or phosphorylation group that's added to that protein. And that's where you're seeing truly differential expression, and you're identifying targets that are only in the tumor and not in the normal systems. Now, the reason that's so important is because this is what directly determines what we call the therapeutic index. How effective is your drug relative to how much adverse event are you going to have from the drug in normal tissues? And that's really the crux of oncologic therapeutics, as well as for other emerging areas. And so the nearest analogy I can use, Danny, is the Hubble telescope, right? When we look up at the sky, we can see a certain number of stars with the naked eye. When we use a land-based telescope, you can see more stars. But when you launch a telescope into space, now you see many, many more stars than you ever could before. And it's not magic. It's just a technology to be able to observe and measure is far greater. And that's fundamentally where we are now with Sapient.
Sapient works across discovery, translational, and clinical development. Could you walk through a use case for each of those to show how a client engages with Sapient, the insights your technology provides, and how this may have accelerated a project? Start with discovery.
Absolutely. And so I'll take this from the theoretical to the actual. And excuse me, Danny, of course, I have to sort of obfuscate some of the details in that you can imagine there's a lot of intellectual property that's the ownership of our clients, and we would never violate that. So I'm going to talk about this a little bit less abstractly, but you'll understand the concepts very easily. So the first question from a discovery approach may be, what is the best target for my drug? And what's happened over the last decade, given the advances in chemistry, is that what we call the druggable genome or proteome, those targets that we can actually engage with therapeutics has expanded massively. It's gone from drugs that have an active binding pocket that are typically enzymatic reactions, such as kinases and other types of enzymes, to now virtually any protein in the proteome that can be engaged by a PROTAC or a biologic drug or a molecular glue or an ADC, et cetera, et cetera. So the space of things that we can drug is far greater. And so the question comes, what is the best target? And what we've done for a number of our clients is we've gone into their tumor of interest. We've measured all the proteins that are altered in that tumor relative to not only the normal counterpart, so not looking at prostate cancer versus normal prostate, but prostate cancer versus all normal human tissues. We've been able to define, hey, these are the actual protein targets that are altered. These are the specific protein isoforms that are altered in there. These are the ones that are most differential. We've been able to identify those that are particularly localized to the cell surface and represent ideal drug targets for emerging therapeutics, such as ADCs and T-cell engagers. And then we've been able to assist our clients in developing the therapeutic that actually targets that specific protein. That's the first step. Now, the second step is once we have that information in hand, we have to be able to develop what we call the targeted assays, where we no longer need to measure 12, 13,000 proteins, but we want to measure one or two proteins really, really precisely. and to do that in a very, very sensitive, very accurate, very precise manner. And so we can develop these targeted assays very rapidly, and that allows us to advance the program into the clinic. Now, once we're in preclinical systems or in humans and early studies, we can begin to ask using these technologies, is the drug that was developed, once it's injected again into an animal or a person, is it doing what it's supposed to be doing? Is the drug actually binding the target protein in that tumor cell? What percentage of tumors actually express the drug? Do we see the expression of the particular target as being predictive of who's going to respond or not respond? And so this is a very sort of definitive and less theoretical way in which you can go from discovery protein measurements or multiomics to identification of a key target, to development of a therapeutic, to early preclinical translational studies, and ultimately using that same assay now in the clinic as part of the clinical studies.
How about in terms of translational work?
Yeah, absolutely. So this is one of the real questions. How do we engage in translation? And we spent quite a bit of time thinking through this. And if you look at many of the early biomarker studies, there's this large chasm between discovery and clinic, and that chasm is marked by translation, where so many biomarkers fall off, so many studies into these multi-omics. They're great for discovery, but then they fail as we transition to clinical studies. And so one of the key questions we've contended with here is, as a service organization, and we operate as a fee-for-service CRO, how do we enable our clients to translate from early discovery to clinic? And what we've built internally here, Danny, is two things. One, a system in which, with our mass spectrometry instruments, we can very quickly go from a discovery assay where we're measuring thousands of things, identify the key target, the key PD marker, the key target engagement marker, and then we can technically translate it to a targeted assay using the same technology. So it's not the case that we have to make the observation or the discovery using one technology and then start from scratch as we want to go into the clinic and find a completely different technology to measure that same molecule. And so that's one way we accelerate it. The other way we accelerate this translational chasm is through data. And so Sapient has built some very unique internal data assets. We call this dynamic IQ. And this dynamic database represents tens of thousands of biological specimens that we've collected ourselves from our academic partners, whether they be blood samples, CSF, saliva, urine, tumor samples, tissue samples, et cetera, et cetera. And what this very large, extremely rich database enables us to do is to make the multi-omics measurements in these tens of thousands of samples, have all the clinical information there. And what we can do with this is we can take a client's data set that may be in a mouse model or a non-human primate, or may still be at a cellular stage, and say, this is your key target. This is your key biomarker for enrollment of patients. Now, how is this biomarker going to act in patients even before you've gone into the clinic and done a single experiment in humans? And this database allows us to answer that question very quickly in silico using existing experimental data. And this allows us then to say, okay, well, there may be three options for biomarkers here. This is going to be the best one in people. Let's advance that one very quickly to development of a regulated assay and use that for your clinical study. And so these are all tools and techniques, both from a technology perspective, from a measurement perspective, and from a data perspective, that allow us to bridge that translational gap between discovery and clinic.
And is there a good clinical example?
Yeah, there's about a dozen examples we have of this where we've made a discovery for a patient. For a client, typically a large pharma company or an early biotech. We've been able to validate that internally with them. They've developed a therapeutic around this. We've developed a target engagement and PD marker. And this is advancing within the clinic to sort of the FDA and beyond. You'll be hearing about a number of these over the ensuing year. Of course, this is the data and ownership of our clients, but a number of these are going to be published very shortly. And so you'll see some examples. And unfortunately, you'll have to invite me back in a year or so here, and I'd be happy, Danny, to review all of these with you as they come to light.
You mentioned you're operating on a fee-for-service model, but is there, do you do this as software as a service? Do you do the work yourself and return the results to a client? How exactly does that work?
Yeah, it's a little mix of all of the above. And so let me walk you through that process, Danny. We do not do drug development ourselves, but rather our mission in life here is to engage those individuals who are doing the hard work of drug development and enable them to accelerate that process. Not only developing drugs faster, but drugs that are more effective once they arrive in patients. And so the way we do that, as you mentioned, is a fee for service operation, and this allows us to be completely unbiased and agnostic. I don't particularly love the term CRO because that denotes a relationship in which you're sending us samples, we're sending you data, and that's the end of the relationship. Typically, the way we're acting is what we call a discovery partner. And that allows us to really engage our clients to simply serve as an extension of their internal groups, to help set them up the best experimental design, to understand the best samples to run. We bring those samples in house. We generate that data using the mass spectrometers and other types of technologies that we have here in house at Sapient. We QC that data. And then we perform deep statistical analysis of that data using our internal biocomputational team, AI based software, other platforms we have internally here. We are using that information then to answer a key biological question. We return all of that information, not only the answer to the question, but all the data that went into answering that question back to our clients. It's their ownership and their property. We can use our internal data assets once again to translate some of these discoveries for our clients. Once again, that's their ownership. And then we can go through the process of developing clinical assays, GCLP or CAP-CLIA based assays that our clients can then use as part of their subsequent clinical trials. And so we work across that pipeline. Our data generation, as you can imagine, is done on a very straightforward fee for service basis. Our statistical analyses are typically done on an FTE basis. Our data asset utilization, which you can think of as a SaaS model, is typically done on a project basis. And so we engage a number of these types of economic models to really provide maximal flexibility and maximal efficiency for our clients.
Many companies talk about AI and drug discovery, but there's still a fair bit of skepticism about it being able to deliver on its problem. What deliver on its promise? What do you think separates hype from meaningful progress?
Yeah, and this is always the path, if you will, around any technological advance. There's typically a lot of hype up front and a lot of excitement. Um, as you then enter into the world of reality, some of that excitement falls away. I think AI has enormous promise, but again, it's fundamentally a tool, and a tool is only as effective as its application. Today, AI based approaches, again, are largely being used to solve only one piece of the drug development problem. And that's around how to identify the best chemical entity to engage my target. But there's many steps before that, and there's many steps after that for which AI is not being applied. And I think this is where the great promise of the system lies, in that it's clear that AI, together with the right multi-omics data, can greatly inform in those other steps. And if we begin to use these systems now, not only for identifying the best chemical, but upfront identifying the best target and validating it, and then using it for the best chemical entity, and then using it for understanding translation, and then using it for the best patient population into which to enter that drug, that's where you start seeing the needle really moving. And I think we're very, very close to that point. As I mentioned, the two factors that you need are, one, the underlying computational systems, the AI networks, and those are largely established. The computing power to do that exists. And now we need the best data. Remember, AI is only as good as the data that is fed into it. And we finally have the robust multi-omics measurements available that we can use for these AI analyses. And we're seeing a number of advances, once again, in target identification and chemical screening and chemical engagers, in translation and preclinical systems, and then ultimately in patient stratification and companion diagnostics. And I think over the next 2 to 3 years, Danny, you're really going to see an acceleration in AI derived therapeutics. And when I say that, it means drugs that are not developed using AI only for chemical screening and for identifying the chemical entity, but also drugs that are more importantly leveraging AI for the entirety of that process, from early target ID through chemical screening and identification in SAR, all the way through translation and clinical studies.
And what do you think the potential here is to use this technology to expand the druggable space by targeting proteins previously thought too challenging to target?
Yeah, there's been so many advances in sort of the druggable space and in druggable modalities. And you can think about this, just a decade and a half ago, the vast majority of drugs were small molecules. And then the introduction of biologic therapeutics and how that transformed the systems. And if you think about where we are now, we've gone from small molecule to biologics to gene and cell therapies to PROTACs to ADCs to T cell engagers to molecular glues to other emerging therapeutics. It's expanded so greatly that the real question is no longer what's the best drug modality or what's can I actually drug this target? The question really now is what's the best target and then what's the best patient? And so I think AI will continue to improve the underlying druggable genome, but I think there's already so many options in that space. If you give a drug company the optimal target under almost most circumstances, they can figure out the best way to drug that target. So I don't view drugging of the target as the big bottleneck. In certain circumstances, yes, it still is a bottleneck, but for the vast majority of proteins, even very hard targets such as KRAS, et cetera, there's a number of drugs are being developed or have been developed that can accurately target that can accurately engage these targets. The big question once again is, what's the best target? What's the best patient? How do I know that the patient's going to respond even before they receive the therapeutic? Those are the key questions where I think AI is going to have enormous impact.
And looking out 5 years, how do you see AI reshaping not only drug discovery, but the way new therapies reach patients? And I guess the ultimate question, does this technology finally move the needle on the cost of drug development?
Yeah, absolutely. It's a great question, Danny. Once again, solving only one part of a complex system typically doesn't move the system as a whole. And so to your point, there's a number of different aspects that have to change here. One, we have to be able to better diagnose patients. And when I say that, have more accurate diagnosis, be able to understand molecular pathology. So not only that you have cancer or that you have heart disease, but you have heart disease, and this is why you have heart disease. This is the actual part of the system that's altered in your specific body that allowed you to develop heart disease. And so that's really a diagnostic issue. We talked about drug development, certainly, and how AI is going to be critical in that process. And then on the back end, you have to be able to match these two things together. You need to be able to understand a patient's disease process at a level that begins to allow you to match the optimal therapeutic to that individual. And this is, of course, the promise and the idea of precision medicine. It's disappointing that it's taken this long to get here, but that's the nature of complex biology and complex specimens. As to whether or not this will move the needle, Danny, I fundamentally don't believe we have a choice. It's clear that the current healthcare system, for a variety of different reasons, is not sustainable, both from a financial perspective as well as a logistics perspective. And there is an absolute need to develop drugs faster, cheaper, that are more effective. And health is one of the basic human principles that's unifies us all, irrespective of where you live in the world and who you are, and what you believe and how old you are. I mean, being healthy is fundamental to the human process, and it's critical that we now develop these drugs that allow us to sort of promote that human health.
Mo Jain, Chief Scientific Officer of Sapient. Mo, thanks so much for your time today.
Thank you so much, Danny. It's really been a pleasure. For any of your audience that's interested in hearing more, I would please ask you to engage us and come find us. You can find us on social media under Sapient Bio. You can find our website under www.sapient.bio, B-I-O, SAPIENT.bio. please email us. We'd love to sort of engage in your listener base. And thank you so much for your time today, Danny.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.